Going to talk about carbohydrates and blood antigen groups. So at the end of this video, you should be able to describe the basic structure and function of monosaccharides, differentiate D and L configurations, explain the biochemical cause and significance of galactosemia, describe the role of amino sugars and oligosaccharides, analyze how glycoproteins and glycolipids form the structural basis of ABO and Rh antigens, and explain the genetic basis of ABO and Rh blood group expression. Now let's do a quick recall from our previous lesson. Remember, we said that carbohydrates are biomolecules that are made up of carbon, hydrogen, and oxygen in about a 1:2:1 ratio. That's why their name literally means carbon plus water. We also call them sugars and starches in everyday language. Now, the main function of carbohydrates is to provide immediate energy. This is especially true for monosaccharides, the simplest form of carbs. They are quickly absorbed and used by our cells for fuel. But carbohydrates aren't only about energy. They also have structural roles, especially in plants. For example, cellulose. Cellulose is a complex carbohydrate that forms the rigid cell walls in plants. And starch is a storage form of energy in plant tissues. So they are polysaccharides. Carbohydrates also have a very important role in communication. For instance, oligosaccharides are found on the surfaces of cells. Remember, they act like ID tags. So they enable cell recognition. So this is how our immune system knows which cells belongs to us and which don't. So carbohydrates are much more than just sugar for energy. They're essential for structure, storage, and cell-to-cell communication. And with that in mind, let's dive deeper into monosaccharides. So mono means one, saccharide means sugar. So monosaccharides are the simplest types of carbohydrates. They follow the formula CH2ON, where N is the number of carbon atoms. Most common monosaccharides have 3 to 7 carbons. And a key feature that they have is the OH group. The OH groups in monosaccharides make them polar and soluble in water. This is very important because it means they can travel easily in our bloodstream and be used for quick energy. Now, you can see here two drawings. So the first one on the left is what we call a Fischer projection. Okay? So the Fischer projection shows the sugar in its open chain form. This is helpful for seeing the arrangement of atoms. It emphasizes the exact arrangement of hydroxyl groups or OH groups on each carbon. So this one on the right is called a Hayward projection. So the Hayward projection shows the ring form that sugars often take in water. Now it's good to know both, but don't worry, we'll focus on understanding the basics. So we can classify monosaccharides in two types. The first one is based on the functional group. Functional groups are groups of atoms or small molecules that give specific property to a molecule or to a substance. So if they have an aldehyde group, so as you can see here, this is an aldehyde. So if you have a C double bonded to O attached to a hydrogen, that's an aldehyde. If you have a C double bonded O and attached to two carbon atoms or the R chain or the side chain, that will be a ketone. Monosaccharides that has an aldehyde is called an aldose. Here, C double bond O, hydrogen. Aldose, the suffix "-ose" means sugar, so aldehyde, sugar. Monosaccharides that has a ketone group, so C double bond O attached to two carbon atoms, is called a ketose. Ketose, ketone, sugar. Okay? This classification is important because functional groups affects how sugar reacts in our body and how it can form rings. So for example, ketoses often form 5-membered rings, or what we call a furanose, while aldoses usually form 6-membered rings, called a pyranose. So this influences how enzymes recognize and process them. So this is important because it helps us predict reactivity, biological role, and behavior in diagnostic tests. Now, the other way to classify monosaccharides is by the number of carbons they have. For example, you have 3 carbon atoms. That is what we call a triose or triose. 4 carbons means tetrose. 5 carbons means pentose. This is important because the carbon count helps define the sugar's role in biology. For example, trioses have 3 carbon atoms and are involved in metabolic pathways like glycolysis. Different pathways in our body use specific sugars with specific number of carbon atoms. Pentoses, 5 carbon atoms. These sugars are important because ribose and deoxyribose form the backbone of RNA and DNA. So they are pentoses. Basically, without pentoses, there's no genetic material. It also determines the structure of sugar and how it connects to other molecules. So knowing the carbon count helps us predict its function and behavior in the body. So this slide shows you the common monosaccharides and their role. Glucose is the body's primary energy source. It's what cells use to make ATP, the energy currency of life. So animals store this as glycogen. Fructose is the sweetest natural sugar found in fruits and honey. So it's sweeter than glucose, that's why it's popular in sweeteners. It's processed mainly in the liver, which turns it into glucose or stores it. Now, you don't usually find galactose by itself. It is usually combined with glucose to make a lactose. So lactose is a disaccharide. This is your sugar milk or milk sugar. So once digested, the body can convert galactose to a different form of glucose, but technically a glucose for energy. So these monosaccharides might look similar chemically, but they have different roles in metabolism and nutrition. So they are essential building blocks for energy and for making complex carbohydrates. So when you think about carbohydrates, not just sugar or like in a negative way, but think of vital molecules that power your cells. Now that we understand the basic structure of monosaccharides, let's talk about another really important detail, their 3D orientation in space. Now, monosaccharides can exist in 2 forms, the D and the L configuration. You can imagine this like your right and left hands. So your right and left hands, they are mirror images of each other, but not exactly the same. You can't lay your right hand perfectly over your left hand. That's exactly what these sugar molecules are like. So this property is called stereochemistry. It describes how atoms are arranged in 3D space. So even if 2 molecules have the same formula, their 3D arrangement can make them very different biologically. Why does this matter? Because our body's enzymes are extremely picky, very selective. They are what we call stereospecific, meaning they only work with one form. Now, for sugars, our enzymes are designed to use the D isomers or the D sugars. Those are the biologically active forms we can digest and metabolize for energy. L isomers, on the other hand, are rare in nature and usually can't be processed by our enzymes at all. Here's an example. For example, glucose. So glucose, this is a D-glucose, this is an L-glucose. What you have to do is to look at the farthest chiral carbon, okay? More of that soon in organic chemistry. But when we say chiral carbon, it's the carbon that has no similar or no the same attachments. So if the hydroxyl group on the chiral carbon is on the right, that is a D-glucose. If the hydroxyl group of the farthest carbon atom is on the left side, that is an L-glucose. L for left. So even though these forms are mirror images, that small difference in 3D shape can decide whether a sugar is useful or something our body can't use at all. Now, let's see why understanding these differences matter, even when things go wrong in metabolism. So sometimes the body can't properly metabolize certain sugars because it's missing a critical enzyme. A serious example of this is galactosemia. Galactosemia from the word galactose, so there is a galactose. Semia, the suffix means in the blood. So you have galactose in the blood. Galactosemia is a genetic disorder where the body cannot break down galactose, which is one of the sugars that comes from lactose in milk. So this happens because of a deficiency in one of the enzymes needed to process galactose. So without that enzyme, or the GALT, or the galactose 1-phosphate uridyl transferase, galactose and its byproducts build up in the body. But instead of being used for energy, that buildup becomes toxic. So it's damaging important organs like the liver, the eyes, the brain. So here on the picture, you can see some of the symptoms like swelling of the brain, loss of appetite, spitting up, or vomiting, jaundice. This is a very powerful example of why proper sugar metabolism matters. Okay? So even though sugars are a critical source of energy, the body has to process them correctly to keep us healthy. Now, under normal conditions, our body has a specific process for handling galactose safely. So in a normal functioning enzyme or normal body, so if you drink milk, the lactose or the milk sugar goes into the liver. So the lactose from milk is broken down into glucose and galactose. So glucose, there's no problem with it because it's readily usable for the body. But galactose is not. So what would happen is that there will be an enzyme called galactokinase or the GALK, G-A-L-K. This galactokinase will convert galactose into galactose 1-phosphate. So the galactose 1-phosphate is turned to a usable form of glucose called UDP glucose. This is done by the work of the GALT, G-A-L-T, the galactose 1-phosphate uridyl transferase. So this is the usable form for the body. In galactosemia, one of these enzymes, usually the GALT, this one, galactose 1-phosphate uridyl transferase, is missing or defective. This means galactose and its phosphorylated forms accumulate, being toxic. So this buildup is what damages organs and causes the symptoms of galactosemia. It really shows how even one broken step in metabolism can have serious consequences for health. So far, we've been talking about basic sugars, but sometimes sugars are chemically modified to do special jobs in the body. A good example would be the amino sugars. Amino sugars are sugars where one of the OH groups, for example glucose here, is replaced by an amino group, NH2. So this small change gives the sugar new properties. Amino sugars are important in structure and cell communication. For example, they're found in cartilage, connective tissues, and cell walls. So this one, when glucose is modified, or if the hydroxyl group is turned or changed into NH2 amino group, it will become a glucosamine. So sometimes this glucosamine is still modified where an acetyl group is added to it via the acetylation process. This glucosamine becomes N-acetylglucosamine. So here are the common amino sugars and what they do. Glucose, as you all know, basic energy sugar. Glucosamine is modified with amino group found in cartilage on connective tissues. This is the reason why you often see it in joint health supplements. N-acetylglucosamine is a further form. It's the building block in chitin, which insects and crustaceans use for shells. So it is also used in glycoproteins that help cells recognize each other. So even though these all start from glucose, small chemical changes make them specialized for different roles in the body. So take note, a simple change in structure can lead to a big change in function. And that's a key theme in biochemistry. Now we're done with monosaccharides. Let's talk about oligosaccharides. These are slightly more complex than monosaccharides, but basically they are short chains of sugars, typically 3 to 10 units long, that are attached to other biomolecules like proteins, lipids. So what do? One of their most important roles is acting like the name tag for cells. So for example, if you wear an ID, your ID is your tag, your oligosaccharide. So let's see how these oligosaccharides are attached and what their roles are in different molecules. So in glycoprotein, when an oligosaccharide is attached to a protein, you will have a glycoprotein. So this works in cell signaling and communication, especially immune response and hormone recognition. Now, when the oligosaccharide is attached to a lipid, it will become a glycolipid. This works well for cell recognition. They also help maintain membrane stability and integrity. So now, let's see how these concepts connect to blood chemistry and what doctors usually test for in the blood. When we talk about blood chemistry, we're looking at the chemical components in your blood that reflect your health and how your body is functioning. So doctors use blood chemistry to check for many things. For example, sugar levels like glucose. They help diagnose or monitor diabetes. Fat levels or lipids to assess heart disease risk. Enzyme levels check for organ function or damage. And proteins for immunity. So they also help in blood clotting. So some key chemistry components you'll see: carbohydrates, basically they are the blood type antigens. So this will help determine compatibility or transfusion. Lipids, they form the cell membranes and anchor important markers. Proteins, like antibodies, they fight infections and hemoglobin that carries oxygen. Enzymes, they speed up chemical reactions in the body. So now let's first talk about the carbohydrates. Blood chemistry is interesting because it's one place where carbohydrates, lipids, and proteins all work together in the surface of the red blood cell. So oligosaccharides on the red blood cell surface act as a blood group antigen. So these sugar chains are like labels that tell the immune system what type the cell is. Your ABO blood type depends on which sugars are present. So for example, your type A, you have A antigen, and the sugar present is called N-acetylgalactosamine. For your blood type B, you have antigen B, so the name of the sugar is galactose sugar. Type AB has both A and B antigens. Type O has none of these extra sugar antigens. So these differences are tiny on the molecular level, but they're critically important for things like blood transfusions. So in blood transfusion, the wrong match can cause serious immune reactions. These sugars just don't float around. They are actually anchored into the cell membrane. So how do these oligosaccharides or these tags stay on the cell surface? They need an anchor. That's where the lipids come in. Glycolipids are lipids that have sugar chains attached. These glycolipids embed themselves into the cell membrane and present their sugar chains outward. So here, they can be seen by the immune system and the other cells. So this setup helps cell membranes communicate and recognize each other. It's crucial for things like blood type compatibility, immune defense, and cell signaling. Lipids don't just act as anchors though. They help maintain the membrane's fluidity and structure, which is vital for the cell to function properly. Okay, so I want you to think of a person wearing an ID lace and an ID. So this person is the red blood cell. This sugars is the ID. The glycolipid here is the ID lace. It's the one that connects the sugar to the red blood cell. So next would be your antibodies, which are basically proteins. So antibodies are like the body's security system. They detect and attack foreign antigens on red blood cells if they don't match your own type. Here's something really important. Your body does not make antibodies against its own antigens. That would be like attacking yourself. For example, if you have type A blood, you have A antigens on your red blood cell. So you make anti-B antibodies in your plasma. Therefore, if blood type B enters your system, those antibodies will attack it. Why? Because they are anti-B antibodies. They hate B blood type. Type B has the opposite setup. They have B antigens on the cells and they have A antibodies in the plasma. Type AB is special because it has both A and B antigens on the cells but no antibodies in the plasma. That's why AB is called the universal recipient. type O is the reverse. It has no antigens on its red blood cells, but both anti-A and anti-B antibody in its plasma. So now you can see how these antibodies help keep our blood system specific and safe for mixing incompatible types. So let's look at how proteins and enzymes in our body also help define and maintain these blood types. So proteins in blood again do so much more than just antibodies. Other important proteins include albumin, which helps maintain blood osmotic pressure, so it keeps the fluid in the right places in your blood vessels. Fibrinogen, which is essential for clotting, stops bleeding when you're injured. And hemoglobin. They carry oxygen to all your cells. But let's focus on the special proteins that actually determine blood type. Your blood type is partly determined by enzymes in your cells called glycosyltransferases. So glycosyltransferases, these enzymes add specific chains or oligosaccharides to the surface of your red blood cell. So for example, the enzyme A adds an acetylgalactosamine. The B enzyme adds galactose. So if you're O, you don't have an active enzyme. So your blood cells lack these extra sugars. This is why the surface of your red blood cell acts like a name tag for your blood type. So when you get a blood transfusion, it's critical to match blood. So this is to avoid immune reactions. So if the donor's red blood cells have antigens and the recipient antibodies recognized as foreign, the antibodies will attack them. So here's a table showing the different blood types. Now, we're not really just type A, type B, type O, type AB. Something's missing. The positive and the negative. Now let's talk about another important blood antigen, the Rh factor or the rhesus factor. So besides the ABO system, there's another important marker. It's called the rhesus factor. It's controlled by the D gene. So the D gene determines if the D antigen is present. If you have the D or the capital D antigen on your red blood cell, even one, you are Rh positive. And if you don't, you're Rh negative. And why is this important? Because Rh compatibility matters in transfusions and in pregnancy. So for transfusions, you usually match both ABO and Rh, giving Rh positive blood to an Rh negative person. So this can cause immune response. In pregnancy, if an Rh mother carries an Rh positive baby, her body might make anti-Rh antibody. So in future pregnancies with another Rh positive baby, these antibodies can cross the placenta and attack the baby's red blood cell, causing hemolytic disease of the newborn. So this is why Rh compatibility is checked so carefully in medicine. But how do you inherit the Rh type? So the Rh factors follow a simple Mendelian genetics. You inherit one Rh allele from each parent. So when we say allele, this is the alternative form of a gene. So the capital D allele is dominant and produces Rh positive antigen. Small letter d allele is recessive and does not produce the antigen. Now, if you have at least one D or capital D allele or capital D small d allele, you're Rh positive. Only small letter d, like two of them, results in Rh negative. So the inheritance pattern explains why Rh positive is more common. So how do we use this? So we can use the Punnett square. So basically, what you have to do is you will be given the alleles of your parents, and then you just have to put them on the boxes. Later on, combine. So what do I mean by that? You're given capital D and small letter d, capital D and small letter d parents. So just write capital D, small letter d. That's this one. And then you write this, capital D and small letter d. What you have to do next is to combine them. So this and this will combine. That's big letter D. Big, small, so that's DD. Oh, DD, PDD. So you have capital D and small letter d, that's CD. And you have small letter d, small letter d, that's small letters. So how do we interpret this? It means that there is 75% chance that your offspring or your child is Rh positive, and 25% chance that your offspring or your child is Rh negative. Okay, let's have another one. For example, you have D, capital, small, and dt parents. So again, just put this here in the boxes. So that's capital D, small letter d. And this one, small letter d, small letter d. And then you combine d, d, that's dd. Small, 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 small. Big, small, big, small. Small, small, this one. So based on the discussion, what would be the ratio? There will be 50% chance that your child can be positive or negative. So this chart tells us which blood types can safely donate to and receive from other blood types. It's all about matching the ABO blood group and the Rh factors. Now you have a lot of things to consider. Number one, the ABO blood group. So the A, B, and O depend on the antigens on the red blood cell. Type A has A antigen, type B has B antigen, type AB has both A and B antigens, type O has no antigens. And number two is the Rh factor. Rh positive means the D antigen, capital D, is present. Rh means no D antigen is present. Rh negative people can only get Rh negative blood, but Rh positive can get both Rh positive and Rh negative. So we call type O minus as the universal donor. It can give to everyone because it has no A, B, or Rh antigens. So there's no risk of reaction in any recipient. So they can only receive from O minus. So they must avoid all antigens in donated blood. The AB positive is the universal recipient and can receive from everyone because it has A, B, and Rh antigens. So it recognizes all types of self, but it can only give to AB positive. So the key pattern is O negative donors are safest for everyone. AB positive people can receive any blood type. Rh negative people can't receive Rh positive blood, but Rh positive can receive Rh negative. Now question, can Rh blood be given to Rh minus? Not generally safe. If you give Rh blood to someone who is Rh negative, their immune system recognizes the D antigen as foreign. So this can cause them to produce anti-Rh antibodies and can lead to dangerous transfusion reaction. Now, can Rh negative blood be given to Rh positive people? Yes, it is safe. Why? Rh negative blood lacks the D antigen. Rh positive recipients already have the D antigen. But there's nothing really foreign to react to. And that's the end of our lecture.